The NIH Consortium for the Impact of Genetic Variation and Function is making incredible advances towards our understanding of human health and disease. The fields of genetics and genomics are not new, and in fact, almost 25 years ago, we had the first announcement of the draft sequence of the human genome. But that was really just telling us what the letters are in this book of the human genome. And if we continue with that analogy of the genome as a book, once we knew what the 3 billion letters were that made up the human genome, we've spent most of the last 2 decades understand how those letters are arranged into words and sentences that make up the text of our genome, and how those words and letters change across different genetic backgrounds and different individuals. But what we've been missing this entire time is the dictionary to tell us what each of those words actually mean. And it's that function of those words that the IGVF consortium is working towards understanding. You can imagine that if we don't know what the meanings of the words are that make up the text of our genome, it shouldn't be that surprising that we have such challenges with treating some of our worst and most common diseases that have genetic contributions, whether it be neuropsychiatric, neurodegenerative conditions, autoimmune disease, cardiovascular disorders, or cancer. And so really understanding this function and this dictionary that makes up what each of the words in our genome is doing and how that varies across cell types, stages of human development, and in response to our environment and different pharmacologic perturbations is really going to be critical to making those next advances towards understanding how our genetics and our genome contributes to human health and disease. As a recent example of some of the work of the consortium in understanding how our genome contributes to human health and disease, we wanted to explore how the environment of a cell or the material mechanical properties around a tissue affect our genome and how that might be involved in disease progression. The mechanics of a tissue are critically important in numerous human disorders, thinking about things like tumor development in cancer or fibrosis, or even things like stroke or neurodegeneration, and even aging. All of these are diseases or processes in which the mechanics of our tissues change over time. We know that that converges on our cells in their genome to change in gene expressions, but we didn't really understand how and why that takes place, where that happens in our genome, and how those changes in genome structure affect the genes and the levels of those genes that are involved in disease progression. So we put cells in different environments of different mechanical properties and then observed thousands of changes in levels of certain genes and tens of thousands of changes across the genome in the structure of different genomic elements. We then mapped many of those to their target genes that we could then link back to genes and regions of our genome that are associated with complex human diseases like fibrosis. And so we hope that this very detailed mechanistic understanding of how our genome responds to environment and how that is involved in complex disease processes like fibrosis will help lead to new drug targets and new druggable mechanisms in areas of human disease that have otherwise been recalcitrant to many of the existing approaches and modalities like fibrosis.